Hi, everyone. We hope everybody's well, and we hope you enjoyed our first plenary lecture. I'd like to welcome you to session 1, Small Lakes and Ponds as Model Systems for the Analysis of Spatial Processes. This session is being organized, and I apologize already for what I'm gonna do with your names, by Dr. Robert Ptacnik and Zsófia Horváth. Okay, I've been approved. Thank you. Yeah. I'd like to inform that the microphones of the participants will remain initially closed, except for the coordinators and presenters, but questions can be asked anytime on the chat. And I'm also informing you that the session is being recorded. So if any of you have any problem with that, the only thing we can say is that just don't comment on anything. So as I said, this session is being coordinated by Robert and Zsófia, and they'll be in charge of forwarding the questions of the participants to the authors. Thank you guys very much. And now the stage is yours. I'll be here in the background. Me and Irma, we are just in the background if you need anything, okay? Thank you again.
Thank you, Priscilla. Hi, everyone. Warm welcome from me and Robert as well to the presenters of this session, and also to the audience who is now slowly gathering, as I can see it. I think we have definitely an accumulating number of listeners. So this is the first time that we're doing this, so please don't be nervous. We are doing it in a really flexible manner. So as you could see it, all the 6 presentations have been uploaded, so you could, if you were interested in them, you could watch them, you could prepare your questions ahead. But at the same time, we also asked you, it was a little bit, late, like during last week. So yeah, I'm sorry for short notice, but it was kind of a late adjustment in the program, that in those sessions where we didn't have that many presentations, there was this idea that it could be a bit more live and kind of conference feeling. If you can, summarize your project, summarize your presentation in like 2-3 minutes speech or talk. You can use slides, but it's not compulsory. So feel free to do it as you feel the best. So first of all, we will go through all these 6 presentations quickly, and after each of the presentations there will be a possibility to ask questions. So we are gathering all these questions also on the online forum. I just checked it and there was actually none, so perhaps it's easier if anybody now has a question along the way, then please shoot them here, so in the Zoom chat, because then we will surely not miss anything important. Yep. I don't know if you have something to add, Robert, before we start with the first one.
Thanks for the introduction, Zsófia, and welcome from my side as well. Yeah, I agree. I just opened the Moodle form and there are, I think, no questions. So maybe it's easiest if we simply, as you suggest, use the chat. And so we try to keep an overview, and I think I can try to keep track of answering everything that pops up in the chat. And we will just do this as a dynamic process. I mean, maybe we can, I guess we are not that many people. We can also take some questions just like interactively, that people just show up. I want to say something. Yeah, we do that in that way. Yeah.
Yeah, sure. And in the meantime, I actually figured that I made a mistake with the order of the presentations, because there were like 2 different orders, and one on the online forum version and one in the Excel. So when I sent you out these 6 talks, the list of the 6 talks, actually it was according to the online, what is that? So it was this homepage where you can also shoot your questions. And officially now I can see it in the final Excel sheet that this is not really the correct order of the people. So I guess we should then actually move back to this Excel sheet so that if some people want to change between the two sessions, then they can calculate with, like, I don't know, I want to listen to somebody who is at the end, and then they can, you know, switch. So yeah, and in that sense, actually the order of talks should be Eric Mateos Barros, Esteban Ortiz, sorry for mispronouncing your names, Ryan Barros Setúbal, David Cunillera Motkuzi, Patrick Brown, and Irma Carvajal, if I'm not mistaken. So this is how we should actually proceed. Yep. Yeah, sorry for the wrong order in my email. So yeah, with that said, I would like to invite Eric to present his work in a little bit of a short format before we start asking questions.
Hello. Well, I prepared a presentation, a short presentation, and just where.
So you should be able to click on share screen. You should be able to share it. Do you have access to it?
Where is it?
It's at the bottom, right next to participants, chat, share screen, Paul, and well, you'll see a different screen than me, but something like this. There it goes.
Can I initiate?
We can see.
So we make this study because we know that bacteria have great capacities, great dispersal capacities, and we are interested to understand how these organisms react to a context of high isolation. So we use it for this data set of headwater shallow lakes. And we have 60 samples scattered over a large landscape, and we have 4 regions, and in each region we have 50 points to 50 collection points. So we use the variation partitioning to see if the environment fraction or the geographic fraction or the which of them are the best predictor for these organisms. For this, we use the biodiversity to see the difference between sites, not the abundance per se. And we found first that we have high levels of similarity, what was expected, because several works showed that these organisms have this pattern. And it's interesting that when we use the abundance to make the dissimilarity partition, we have minor, how to say, spatial turnover, or sorry, nestedness, and all this, the similarity is because of the spatial turnover. Also, we make the half creek, and we can see that when we use the species identity or the presence absence data set, we have lots of similarity between sites. And for the other hand, when we use the abundance, we have dissimilarity between sites and no significant difference that can be also inferred as the drift acting in these communities. Finally, there are the variation partitioning, and we can see that we have first the geography was the most important fraction. It's explained the major part of our variation. What was surprising, because other studies don't show it, and we also see large unexplained variation that can be doing because a sampling effect. Maybe we don't collected some variable that is impacting this community, this math community. But also we could be seeing a neutral process, what is comparable to the Half Creek results. And we also could see that if we partitionated the environment, the pH is responsible by 1/3 of all the variation for this fraction, which is expected because in other studies we recovered this information. pH is the best environmental predictor for this data. And finally, we can see that in the geography fraction, the regions are the best predictor of variation, which is the result most interesting in my vision, because it's not common that this type of geographical aspects is collected or inferred by bacteria, because we assume that these organisms are capable to overcome all the geographic barriers, and we can see that maybe it's not the case. Some bacteria will be regionally restricted. And it. I think I said all for this study. It's just a resume, an abstract.
Thanks, Eric. I would like to ask if anybody has a question in the audience at the moment, because we don't see anything asked on chat, so you can just feel free to ask in person anyway. I think we are kind of missing this already for a while, all these personal connections, so go ahead and use the opportunity to discuss stuff. Yeah, Robert.
Yeah, it's just an initial question. It seems that it was, can you tell a few words about the effort? I mean, it seems that you covered a large number of habitats actually in quite a big area. I mean, I don't know the environment there at all. I have never been to South America anyway. So can you tell a bit about the sampling effort? In which time did you access all these habitats, and how much time did you spend at the habitats? Just a bit of a background information for us.
This sampling is part of a bigger study that collected 300 samples in different locations, and this was by about 5 years, and we selected these 60 samples to sequence the DNA and make these studies. It's a snapshot, basically.
Thanks. Luke had a question because he raised his hand, I think. Hi, Luke.
Yes, I think. Thanks, Eric, for the very nice study and lecture. So my question was actually the same as David. So I mentioned in the chat so that I could pass it to him, but you picked me out. Sorry for that, David. But you can give a follow-up question. So basically, I think. So you find, Eric, that this pattern is strongest for regions. And so that makes me wonder how the regions differ in their environmental settings, for instance, soil or what. So, I mean, these are relatively large areas, so they might be totally different in the setting. And then it's actually, again, an environmental setting rather than a spatial one. Can you comment on that, please?
But these regions are not very much different, because here the soil is much impacted. It's basically a great farm with sugar cane, and when we selected the samples, we selected an equal number of lakes that present I forget the word. The trophic state, we selected 5 samples that was oligotrophic, and the other 5 that was mesotrophic, and the other 5 that was eutrophic in each region. So in this case, we think that the trophic state will not impact the results, and this probably is because of the distance, because of the neighborhood was also an important geographical aspect, and basically the most important geographic information that becomes of the studies, that the 2 regions that was more distant was those that was more different and impacted more the results.
Okay, thanks very much.
Thanks, Eric. I think we have to move on. So actually, if you have any more questions, then just please go ahead and ask them on the online version, on this online forum page, where you can, basically access it until the end of the conference. So I would also invite all the presenters to regularly check back, because sometimes, it can happen that somebody asks something after your talk, or maybe tomorrow after another talk that just, gives them an idea to ask back. So please go there regularly and check. So moving on to the next presentation of Esteban Ortiz. Hi, Esteban. Please, the stage is yours.
Okay, thank you very much. Let me share my screen. Okay, can you see it? Okay, Hello, everybody. Let me show you a brief summary of my presentation entitled Meta-Community Diversity and Body Size Beyond the Scaling in Dispersal. So, I'm sorry. Okay, in a meta-community context, there are three key elements that affect diversity. They are local density, dispersal, and diversity of the regional pool. These three determinants are highly connected with taxa body size. And the focus of both theoretical and empirical studies has been on the effect of the scaling relationship between dispersal ability with body size on diversity. However, the effect that the interaction between these three scaling relationships has on diversity patterns has been less considered. So our study focuses precisely on this issue. So we evaluated the interconnection between these three scaling relationships with body size and diversity patterns from a theoretical and empirical point of view. First, we used a coalescent-based meta-community model, which simulates local communities by rebuilding the genealogy of individuals in order to obtain the expected relationships between alpha, beta, and gamma diversity with body size. Then we analyzed how diversity scales with body mass in a natural meta-community of temporary ponds, included in the analysis 11 animal orders. And then we compare the expected versus the observed patterns. So in a nutshell, we observe a high congruence between the theoretical and empirical results. That is, we reported a negative relationship between alpha, beta, and gamma diversity with body size. And the take-home message is that this negative relationship between diversity and body size, taxa diversity and taxa body size, is mainly driven by the combination of two scaling relationships. The scaling relationship regarding local density with body size and the scaling relationship between diversity of the regional pool with body size. The second point to highlight is that the scaling relationship between dispersal ability with taxa body size attenuates this negative trend by enabling the coexistence of larger species. So the final point, the final message of our work is that a more comprehensive approach about the role that mass could play on determining meta-community diversity at several scales is needed. So that is the summary.
Thanks, Esteban. Thank you. Very nice talk. I wonder if anybody has a question in the audience. Robert?
Just to make a start, thank you. Very nice talk. I like the idea to address the body size in a systematic way. I was wondering, it's often, though there are a number of studies that report that there is a scaling relationship with trophic position and dispersal, so that organisms in higher trophic level tend to be more dispersal limited. That has been suggested by a number of models. And obviously there is a correlation between body size and trophic position in general. So I was wondering if your study allows also something if trophic position, apart from body size, fits in this pattern that you find? Or did you consider trophic position as well?
Yeah. Okay. No, it doesn't consider trophic position because it doesn't consider any kind of interaction between individuals. It is just a neutral model in which you feel empty spaces, and you run the model until the whole community is filled. But it can be included in the model. Yeah, I mean, it is the next stage of the development of the model, to include two things: the interaction between individuals and the local filters. Because in the model, it is a neutral model. There is no species sorting in the communities. But it is, you get a point there.
So we have another question from Patrick Brown. He raised hand.
Hello. I was wondering if you noticed that if your scaling relationships changed based on the mechanism of dispersal, such as passive versus active dispersers, if that changes the scaling relationships you looked at?
I'm sorry, can you repeat, please?
I was wondering if you noticed if your scaling relationships changed based on the mechanism of dispersal, like passive versus active dispersal.
Okay. We didn't include passive dispersers. We fed the model with body sizes of all active animal dispersers, animal taxa.
All right.
But thinking in the air, yeah, the scaling relationship between dispersal and body size for passive dispersers is the opposite for the active dispersers. So it would, I think it would change the results in some interesting way.
Can I quickly add on this question?
Yes, of course.
Thanks. Regarding size and dispersers, I mean, I think you have flying and non-flying organisms in your, so aren't the flying organisms a group on their own in this data set? And how would the flying versus non-flying fit within the size scaling? Are they evenly distributed, or are the flying organisms on the larger end?
No, the larger organisms are killifish. And yeah, the flying organisms are in the lower part to the middle part of the body size gradient.
Okay. A silly question from an outsider: how do killifish mainly disperse apart from swimming? I mean, they lay eggs in the dry phase, right, or before drying out of the habitat. Is there some dispersal of the eggs of the killifish?
Yeah, we have two things to say. The system is a grassland, a plain grassland. So when it rains a lot, small currents, you can see small currents between ponds. Actually, in fact, you can identify which pond is connected with each other pond. So we think that adult fish could disperse through this stream, this small stream, and we never tested it, but there are cows in the system. So a possibility is that they could transport eggs from pond to pond in the mud stick in the feet. But it is just a crazy or not crazy idea. We didn't test it.
It's an interesting question, because the other fish don't have such a possibility. So I think there was a question from Luc de Meester also.
Yeah, I think that's kind of the last question that we can take now. But yeah, Luc, thanks.
Yes, first of all, thanks very much, Esteban. A really wonderful presentation and study.
Thank you.
I did notice that I would also check a little bit on this passive versus active dispersal, because when I was looking at it went quickly, but I was sort of thinking that both of them are in there and they would have different relationships. So, but my question was about something else. So I think it's very clever to consider both the local numbers and then also regional diversity, and then look at how that impacts dispersal in addition to the real dispersal capacity. So first of all, that's a very good idea, and it's very good that it's explored. I was wondering whether the regional diversity is actually a consequence of the local abundances. And so whether you would be able to sort of see whether there is a double effect of these local abundances, that part of it is through the regional diversity, part is independent. And so that things actually always at the meta-community level become worse and worse for large and less abundant species.
Okay, if I don't misunderstood, can you repeat the question, please?
Yes, I can. So the point, I mean, you mentioned 3 factors, no? And so 2 of them are local abundances, and the other one is regional diversity. But you could argue that regional diversity is largely a consequence of the local abundances.
Yes.
And so that they get spread more, and because they get spread more, there is high regional diversity. But that creates a setting. So first of all, my first question is, could you be able to disentangle that with your model, whether it's a direct or an indirect effect? And secondly, if that would be the case, it would make things actually always worse and worse for larger organisms that are more rare, because they would not only have the lower abundances, but they would have less sources in terms of habitats. So in terms of non-equilibrium meta-communities, you get easier to the point where everything crashes.
Okay, regarding the first question, yes, you can separate the effect of the original pool and the meta-community pool, the dispersal from other communities, because you sample, you can sample an individual from the pool or from the local, the neighboring community. So you can set it as a parameter. You can set, so you can, yeah, you can take into account both effects by isolation or combined. I think that was your first question.
Yeah, absolutely. Yeah.
Okay. I'm really sorry, but I didn't understand completely the second part of your question. I'm sorry, really sorry.
As a comment, I think it's just interesting to reflect on the fact that with respect to conservation and non-equilibrium meta communities, so the ones that would in the end die out, you have this double effect for larger species. And perhaps it's even possible with your model to actually model that, which would be really great if you could do that.
Okay.
But thanks again for the lecture. Thanks.
Thank you, Luke. Thank you very much.
So thank you all. We're moving to the next talk. Thanks, Esteban, once again. So the next one will be Varian Satuba, and she will be with us, yeah, in a moment. So if you want to share a slide, then just go ahead and the stage is yours.
Hello, everything's okay?
Yeah, we can hear you loud and clear.
Okay. Hello, everybody. Thank you for the audience and thank you for the opportunity to show my study here. Wow. In this study, we went to evaluate the functional complementarity of the zooplankton community between temporary ponds and permanent lagoons, given the difference in their hydroperiod dynamics. And several studies have shown that complementarity between different water bodies. And complementarity is an important characteristic to understand biodiversity patterns and can be understood was the property of the set of objects that exists when at least some of the objects in one set different from the object in another set. So an analysis of the community functional complementarity in light of their potential different response to environmental variability allows a better understanding of the ecosystem dynamics. From this, we hypothesized that temporary environments will be functionally more diverse than permanents and will have different functional composition. And for this, we compare 5 temporary ponds and 5 permanent lagoons in relation to their limnological characteristics, species richness, functional diversity indices. Let me see. Oh, no. Okay. So we found that temporary ponds were characterized by very low depth, salinity, and pH values, and high values of chlorophyll A and nutrients. On the other hand, the permanent lagoons were characterized by the low concentration of nutrients in chlorophyll A. So in this PCA, we can see a clear distinction between the 2 types of environments. So in relation to species composition, we found a total of 73 taxa in temporary and permanent environments. Of these, 38 taxa occur exclusively in temporary environments. The values of functional diversity varied among sampling dates and type of environment, but functional diversity and species richness were not significantly different between temporary and permanent environments, except for functional dispersion, in which temporary environments presented higher values than permanent ones. And so the 2 types of environments were also different regarding the composition and abundance of functional traits. Temporary environments showed significantly higher functional expressiveness than the permanent ones in the first and second sampling. The zooplankton community from temporary environments was characterized by functional traits related to omnivorous and carnivorous trophic group, current feeding, scrapper feeding, and coastal habitats. The zooplankton from permanent lagoons was characterized by pelagic habitat, filter feeding mode, and herbivorous feeding preference. So in summary, temporary and permanent environments are equally diverse in terms of species richness and functional diversity. However, the environments differ among them regarding the composition and expression of traits, revealing high functional complementarity. Our results demonstrated that both temporary and permanent environments are heterogeneous and diversified, especially the temporary ponds, where each one presents a unique and diversified set of species and functional traits. For the permanent lagoons, there is an overlapping in functional traits, indicating that in all environments harbor different species and consequently functional traits. The result that lagoons and ponds are functionally complementary was predicted by our initial hypothesis. So our take-home message is temporary and permanent environments present functional complementarity that makes them important to maintain the functional diversity on a regional scale. Is for species that do not arrive or develop in more stable environments, because these species are more likely to colonize environments that are periodically restarting their successional trajectory. And also the temporary ponds are especially important because of their unique characteristics and their role in meta-communities dynamics. Well, so thank you for your attention, and it will be a pleasure to answer your questions.
Thank you. I wonder if anybody has a question? Because I've been checking the forum, but none there. So Robert, please go ahead.
Yeah, just a quick question. I would be curious, has there been a systematic difference in the habitat size between temporary and I mean, the picture you showed suggests that the temporary ponds were maybe rather small and the lagoons were rather large, but maybe that's just due to the pictures. Has there been such a trend?
Yes, there is a little different, but in case of our environment instead of
Connection seems to be very bad.
Temporary, they are large, but very shallow.
Mm-hmm.
Yeah, I was also wondering about the same thing regarding salinity, because I think in your longer slideshow, you actually showed that it's also quite markedly different. So like the, how was it, the lagoons, they were saline, they were next to, I guess it was like an oceanic input from salinity, and then your temporary ponds were actually not saline, if I understood it well. Did I get it right?
I think we lost her.
Maybe, Ryan, try to switch off your camera. Maybe it helps if your connection is not so good. Well, if it doesn't get any better, then yeah, I would invite everyone to write the questions in the homepage format so that she can get them later on. Yeah, I think she lost the connection,
Trying to
sadly.
reconnect now. So maybe we move on to the next talk, and hopefully at the end the session we have some time to talk again.
Okay, so then the next presentation will be from David Cuniera Moncuzi. Hi, David.
Yes, hi. I'm sharing the screen. Let's see. Yeah, can you see it?
It's coming through.
Everything froze, but I think that...
And it's here.
Okay, perfect.
Works.
So, well, just a fast summary of what I presented here that you already showed that, yeah, it's a work that I did during my PhD thesis, which is entitled Meta-community resilience against simulated gradients of wildfires, disturbance intensity, and species dispersal ability determine landscape recover capacity. So just to kind of refresh a little bit what we were talking about it, or what I was dealing with, it's like the interaction between disturbances or wildfires in this case and temporary ponds, as wildfires are probably get to become more intense and stronger in the future, and the interaction or the impacts of wildfires on these systems is not well studied, or it has not received a lot of focus. During my PhD thesis, we did some works at the local scale, analyzing several ponds that were affected by wildfire, but we were still lacking kind of the landscape perspective and also the future scenarios idea in this sense. So in this work, what we tried is to gather what we knew at the empirical level, which was that wildfire mainly affects species in relation to their dispersal capacity, and also that seasonal changes are really important in terms of recovery after wildfire. And we tried to understand how future scenarios, understanding them as increasing intensity and increasing extension of wildfires, and also at the landscape level or at the landscape scale, will affect or will impact Mediterranean temporary ponds. And we use for that simulation approaches to draw these gradients of extension and intensity to explore their impact on community diversity, and also to understand community resilience and their response in future scenarios. So just to be super fast in this sense, we had a reference wildfire, which was one called Empordà wildfire that happened in northeastern Iberian Peninsula that affected several ponds or several water bodies in a region. We translated these water bodies into nodes of a network and connected each one of them using several threshold distances that at the end created several networks or landscapes in this sense that could be somehow related to dispersal capacities, being more disconnected landscapes related to passive dispersers and more connected landscape to active dispersers, for example. And then we also simulated several wildfire scenarios, taking into account these networks and changing their extension and intensity in order to draw somehow different gradients and also future scenarios in this sense. So what we could show at the end is that testing and comparing with the imported wildfire or the original one, what we saw is that our model was indicating or was validating what we had already seen empirically, so that wildfire was affecting mostly to passive dispersers, and also these organisms were the ones having more difficulties to recovery after wildfire. But when we moved to the simulation scenarios, what we saw is that both intensity and extension were not behaving similarly, and instead of a gradient of greater wildfires imply greater impacts on community, we saw that this change was abrupt, and we were passing from a completely recolonized community, which would be represented as blue colors, to a completely impacted community, which would be represented by red colors. And that this change, a part of nonlinear and abrupt, was mostly mediated or related to intensity. At the end, we also saw that our wildfire would be placed in a currently resilient region, which also agrees with what we see in the field, that the recovery was really fast. But this would be in the landscape scale or more in a more regional scale. At the end, if we try to think about future scenarios, what we saw is that if we expect an increase of wildfire regime, so an increase towards the intensity and the area axis in this sense, but also we expect a loss of ponds and fragmentation, what we could expect is that what we now have as a currently resilient system would move in the future towards a currently compromised system. So that at the end, the wildfire that we have observed, although now appears not important or not compromising the community, might be in the future if the tendency is followed in this sense. Having said that, thank you again for your attention, and if you have questions or anything, I can answer them here in the forum or in any other place.
Thanks, David. Is there any question in the audience? I don't see one yet. In the meantime, maybe I have one. I was wondering about the effect on actually kind of the resting stages, if you can say a resting stage also for like snails when they're, not shut themselves down and then, yeah, they are in the sediment and trying to survive all these wildfire events. Were you thinking about including this perhaps in some way into the model? Because maybe it's somehow similar to the approach that you had with the intensity of the wildfire, because I guess that's also kind of related to that the more intense wildfire will maybe even kill the, I don't know, like bacteria, seeds, and perhaps a less intense wildfire will leave, I don't know, the snails alive. Do you think it could be taken into the model?
Now it's not directly included in this model, but of course it's like the kind of the natural following step in this sense, because for now what we assumed when we were burning pond in this model, it was that everything was erased or was impacted in this sense. But of course there is organisms that still survive the wildfire and remain there, and this is especially with resting stages, because at the end most of these organisms are like buried in some sediment of layer that at the end makes them more resistant indirectly to the wildfire. But it can be included, of course, and it's kind of the natural step to account with it, because for sure it determines, of course, super strongly the post-disturbance recovery in this sense, as a way on like priority effects and all these lines in this sense, more than colonization.
Thanks, David. Robert has another question.
Yeah, short question or two. Would the wildfire have an effect on dispersal in the sense that the wildfire, after the wildfire is over, there is, I imagine, more dust because the landscape is more open. The open landscape is more open to wind. And second, does the wildfire affect the fertility of the sites? Does it cause even some fertilization effect due to remineralization?
Yeah, I mean, the impact at the dispersal capacity or of the movement of organisms, for sure it has to have some kind of effect, because at the end the whole region is affected and the wildfire changes at least, I mean, vegetation loss and all this, and maybe after you have more stronger winds. I really don't know how this could be interpreted, but at the same time, regionally speaking, and specifically for this side, winds are already strong, so it would be tricky to really draw how it would work, but for sure it has to have some effect. And in the second question, that was the fertility, yes, yeah, for sure. In fact, in the previous studies that we empirically went to these ponds and test them, we saw that especially at the beginning of the period, once they fill up, they have like of a pulsed effect with an increase in nutrients, chlorophyll, and then some feeding groups even of macroinvertebrates that increase their abundance in these ponds. But it's kind of really pulsed at the beginning of the period. In fact, in lakes and rivers is what you could expect after a wildfire, that at the end nutrients enter in the system, and this triggers kind of the.
I think in historic land use, the burning was also partly for immediate fertilization of the land, right? I think there's some historic land use practice. Yeah. At the end, ash is a nutrient. So and even you could observe it like directly on the ground, kind of, because ponds were like blooming with green after a while. So it was like really kind of a fertilization effect, let's say. But yeah, for sure. But communities or macroinvertebrate communities in this sense respond super fast to that. So you have an increase of nutrients, chlorophyll, algae also, and then you have also a kind of blooming of consumers of this. Thank you. More questions to David?
Yes, I also have one question for David. Sorry, my camera was off. Thanks for the interesting talk, David. And I was curious about the chances that you get a very intense fire, because you showed that the one you had was in the end fell like within the range that it was not very impacting for the community. Mm-hmm. But I think that unfortunately we are having in this region more fires more frequently than it naturally should be. So what's the risk for these ponds, like the current risk, more or less?
Precisely is this word, because all this comes from kind of like we began to study locally how wildfire was impacting, and we were seeing that not much was happening in this end. Like, so the ponds were rapidly recolonized and nothing happened at the level of communities. But because at the end, when you kind of draw these general pictures of different intensities and extensions of wildfire, you see that maybe this wildfire that was big was not that intense. But once you have an increase or once we will have an increase that it's clearly expected and that will become, then we will have the most thing that I think that it can happen at the end is that recovery will take much more time, although maybe the organisms will be able to reach the ponds, but it will take much more time. And also I always kind of couple it with pond loss and fragmentation of the network, which at the end is super important to really use ponds as stepping stones to reach to burned ones. So at the end, this combined effect of an increase of intensity and a loss of ponds is really not pointing to a nice future in which ponds will, and at the end, the ponds will rely on what they keep inside, which is mostly more generalist taxa, and the recovery will be much, it will take longer, or even it will not be feasible in this sense. And you will lose populations of snails in this case, or undetermined groups that will not be able to reach the ponds and recolonize them. after an increase of frequency also and intensity. So yeah, precisely it's the interesting part, I think, of all this, that at the end is, you see that they are resilient now, but in the future, this tendency or this strong pattern that you see will not be maintained.
Thank you very much, David.
No, thank you.
Thanks. In the meantime, Ryan, are you with us now? Because we can also take questions addressed to her if.
Yes, I'm here,
yeah,
but I will turn off my video to.
Fine.
No. Okay.
Yeah, go ahead. Don't worry. So we have 5 more minutes before we move on to the next talk. So anybody who has a question either to David or Ryan, please go ahead.
I had a question to Ryan, if we are still in time. That I wanted to, now that I'm here. The thing is, are these ponds either the permanent ones or the temporary connected somehow with ditches or something? Because I saw that in the map that you presented, some of them are quite close, and this might drive some of the similarities or things that you were, or the non-differences that sometimes you were seeing. It's just out of curiosity. Did you hear me?
Hello?
Hello. Could you hear me, Rayana?
Yeah.
Ah, okay.
And you?
I am hearing you now.
Okay. Well, I'm saying that the difference is the interesting point in the study, because it's not can be justified by the difference in the space or local factor, because they are the same. But I think we hypothesize that these differences are because the ponds restart their successional trajectory, and then this kind of environment have a different taxa because of that.
Thank you. I think it was a bit of interference from, at least from my point of view, but
can you hear me? Okay.
Yeah, now we hear you, but then at some point it cuts. But don't worry, it was just out of curiosity. I understand that there is no such connections. So perfect.
So yeah, and when we got disconnected, I was actually asking the question regarding salinity. So in your longer talk, you referred to salinity as kind of a big difference between the lagoons and the temporary ponds, if I remember well. And I was just asking you if this can drive any of the differences that you find, because this is a really serious environmental stressor. So of course it can lead to very different communities, even if you had exactly the same type of habitats, so like lagoon versus lagoon.
Yes. The salinity is an important factor for the coastal lagoons because they are very close to the sea, and then they are influenced by the marine spray and other effects about the salinity. But the ponds are also influenced by the hydroperiod during the desiccation phase of the hydroperiod. Then the salinity is also important for ponds.
Okay, thanks. Any more questions, either to David or to Ryan? No? If not, then perhaps we can slowly move to the next, the 5th talk, I think. So that's supposed to be Patrick Brown. Patrick, are you with us? Yeah, hi. Thanks.
Hey,
then the stage is yours then.
Can you hear me?
Yep.
All right. So the sun just came up here, which is pretty Exciting. Let me share my screen and I can get started. Let me see.
What's the time there?
It's 7:29 a.m.
Gosh, that's early. Good morning then.
All right. The project was called Interpreting Priority Effects in Shallow Land Arid Waters, but unfortunately I think we're gonna have to change the title to something more like Interpreting Stochastic versus Deterministic Effects in Shallow Land Arid Waters, since the due date for the manuscript is kind of creeping up on us and we're not done all our analyses yet. So what we were doing was we're looking at a subset of data from a previous study we published on water systems in the Chihuahuan Desert. We're specifically looking at rock pools and playas and tanks, these systems in the Chihuahuan Desert. And you can see here, I have them kind of mapped out. Can you see my screen? Okay. The playas are the blue circles, the tanks are the red squares, and the rock pools are the purple triangles. So we have them from a range of areas in the Chihuahuan Desert. We're interested in looking at how, what's structuring the species assemblages in these habitats. So we sampled 60 rock pools, 17 playas, and 14 tanks over a period of 20 or so years. I didn't do the sampling myself. It was done by multiple people in that period. And we have between 1 and 20 sampling events per site. So there's a really high difference in the sampling effort at each site. And we sampled both planktonic and littoral rotifers. And we also measured select physical parameters at every site, including latitude, longitude, habitat type, hydroperiod, and some proxies of productivity. And we identified rotifers and created species assemblages from this data. And here's kind of what our results look like. So for species richness, we found the lowest richness in rock pools and the highest richness in playas. And we did a canonical correspondence analysis to look at the impact of our environmental factors on this data. And we can see that from this, the hydroperiod appears to be very important. It's along the first component. And also the conductivity was very important. And out here along the conductivity related to, we have rotifers that are traditionally associated with saline environments, such as Brachionis plicatilus. When we had all sites included in our data, someone has their microphone on. All right.
Yes, I believe it's Jorge. If he can turn off his microphone for a bit, please.
Sorry. So when we looked at area versus species richness, we found for all our sites included that there was a relationship. However, our different habitat types have very different sizes. Playas are much bigger than rock pools. And it turns out that this relationship was probably just due to the difference in habitat type, because when we look at each habitat type separately, we don't see this relationship between size and species richness. We created per simper profiles to look at the influence of stochastic versus deterministic effects in structuring our species assemblages. And from these profiles, we found that our empirical data, these red circles on the left-hand graph, most closely matches the stochastic models, which implies that dispersal is important in structuring our community assemblages. We found that certain species are associated with certain habitat types, such as Hexarthra and rock pools. And from our per simper analysis, we calculated this dispersal niche continuum index, which should show the influence of deterministic versus stochastic effects in structuring our species assemblages. And a negative dispersal niche continuum index indicates the prevalence of stochastic effects. And that's what we found for all of our site comparisons. The less negative it is, the more deterministic effects should matter. And this is an interesting index. It hasn't been used in many studies. One thing I'm curious about is how nestedness influences this index versus species turnover. I'm not sure. It's something we want to look at. And we wanted to look at this data in terms of priority effects. We haven't quite got there yet. We've mostly looked at the impact of stochastic versus deterministic effects on our data. So if you guys have any ideas how to tease out the influence of priority effects versus pure dispersal, I'd like to hear it. And I guess if you have any questions, I'll take them now.
Thanks, Patrick. Do we have any questions in the audience? No, not yet. Then maybe I will start with a simple one. Perhaps can you exit your slideshow, please, so that we can see all the participants in case, yeah, somebody's raising a hand or so. Thanks. A simple question. I was just thinking about these tanks versus the playas. So in the plenary talk now, we learned that, yeah, in some parts of the US, you call the lakes or shallow lakes tanks. But then what is actually the difference between tanks and the playas?
All right, so generally tanks are man-made structures that are created to feed water for cattle and ranching. So something's named a tank.
Okay, so it's a different tank then.
Yeah, there's probably different definitions in different areas.
Okay.
But most of what we're calling tanks are cattle tanks specifically. So in our study, we use tank to refer to the artificially created ones, and we use playa to refer to the natural habitats.
Right.
Some tanks may have originally been natural playas that were altered to supply water to cattle.
So in the meantime, Luke has a question.
Yes, Patrick, thanks for your nice talk. I was, of course, stimulated to be very attentive because the word priority effects was in there, and then you did not analyze that yet. So with respect to how to analyze it, I thought that you had, I have two suggestions perhaps, I mean, I do not know. I thought you had mentioned that you had some temporal data, so that if that is the case, I mean, I do not know. I mean, you said it was sampled across different periods, but I do not know whether you have repeated samples. Huh? So that's one thing. The second would be, one way to check that would be that you would check whether these dispersal effects or these stochastic effects that you find are stronger for ecologically more similar species, because they would have more, would be more subject to priority effects, whereas the ones that are clearly different in environment would not have any priority effects. So I do not know whether there's any possibility to disentangle that. I mean, it's just an idea that popped up now. I have never tried to do that in our own data sets. So, but perhaps this is a way to go. Just a suggestion.
I think, yeah, I think we might be able to do that. We can look at their ecology. We have a good idea of what most of them eat and how they live. So we might be able to look at it that way. Thank you.
You're welcome.
Any more questions? So maybe a quick comment or suggestion from me, and then Robert. So like when you showed us the species richness values, and it was especially rather in the longer version of the talk, you showed us these box plots for the 3 different type of habitats. And I was wondering if you looked at it from the sampling scaling point of view, because I observed that you had much more rock pools compared to, for example, playas, and then it means you cannot really directly, you know, compare them with a simple box plot or an over. I don't know how this figure was, but you should take into consideration that you had a different sampling effect. So you could tease that apart either with a species accumulation curve, with an increasing number of samples, and then you can see it if it's indeed a different species richness in the 3 different types of habitats. Or you can also go for some permutation-based comparison. So yeah, if you want to really efficiently compare the species richness values, then maybe dig a little bit deeper into this. That's just a suggestion from me. And then Robert had a question.
Yeah, thanks for the very nice talk also from my side. Yeah, regarding what Zsófia said, also related to this, I don't know, yes, we didn't talk about the sampling protocol really, but I think that might also be, you might have differences how representative your samples are in the different systems, also depending how well they are mixed. That could also be relevant to consider. But my question would actually be, if these tanks are specifically associated with cattle, so for providing drinking water, I would wonder if how this, in fact, practically associated with cattle. So would you expect that these systems potentially are, could be affected in terms of dispersal facilitated by cattle or something like that? Is it relevant in these systems?
So most of these were built to water cattle, but they're no longer active ranches. So that might be a historical effect. I know that some other people in our lab group are looking at the impacts of feces on rock pools, but not from cattle, from Barbary sheep. So I'll be interesting to see how that turns out. I'm not doing that though. But we do expect to see impacts from large mammals on our rock pools and tanks.
Okay, thank you. Any more questions to Patrick?
Yeah, well, if none, then perhaps we can also, no questions. Okay, then thanks, Patrick. And then let's move to the last talk of this session, if I'm not mistaken. And this will be given by Irma Carvajal.
Yeah. I will share my screen. 1 minute, please.
Thank you.
Everybody can see?
Not yet. Yeah. Now it's
okay.
There. Go ahead.
1 minute. Okay, my name is Irma. I will present my overview about my results. And the title is Effect of Natural Substrate, Environment and Spatial Factors on Alpha and Beta Functional Diversity of Phtacnik algae. So macrophytes could increase the habitat complexity because of our diverse niches and may also affect diversity partners. And the difference between Cara and Nitella, Cara and Nitella have a difference, morphological difference, and this could increase the partners of Phtacnik algae. So because of this, we tested the hypothesis that the substrate type, Cara or Nitella, could provide different microhabitats to Phtacnik algae, increasing the phyton alpha and beta functional diversities. Being a local filter that explains better modification partners of Phtacnik diversity than environmental and spatial drugs. For this reason, for this reason, for answer this hypothesis, we collected the 19 man-made shallow lakes, and we measured abiotic and biotic data. Phtacnik traits were classified into four traits and 18 categories. And we used this information for calculated alpha and beta functional diversity. In summary, we found that the functional richness, evenness, and divergence were similar in substrate type. The functional richness and evenness were different between the lakes with Cara and Nitella coexisting in the same lake and Cara and Nitella in lakes isolated. And F-richness was influenced by abiotic and biomass substrate. The beta functional diversity, yeah, we found that the variables environment and spatial was homogeneity, and the beta functional and taxonomic were similar for both substrates. So in conclusion, we agree. Sorry. The complementarity effect explain the coexistence between both communities, and the closeness between the lakes demonstrate a scale effect, and the species shorting being a main mechanisms behind meta-community epiphytic. So the environment and spatial drivers were the most important for explaining the alpha and beta functional diversity of epiphytic algae. So thank you for attention and for patience.
Thanks, Irma, also for being so short and on spot with the summary. Are there any questions in the audience? If not, then I can ask something. It's a simple one. So I was wondering, when you said that there was actually a difference regarding functional traits, whether a community was, you know, assembled on a Cara or on the other plant. Can you recall like what actually was or were these kind of functional traits that differed among the different algae? So was it related to how they get attached to these plants, or was it something different?
Yeah, we use 4 traits in 18 categories, like a size, productive, life form, if we're unicellular, I don't remember the exact term in English, sorry. But attachment form too, if the algae were loosely or firmly, you know. And I believe that the Cara, that is more complex, will support more traits and more structures like loosely attachment than others. I don't know if I answered, but it's like this.
Fine, thanks. And I was also wondering if you know something about the possible difference on like micrograzers that might be, you know, moving along the branches of these algae, that perhaps the micrograzers might be different. Do you have any knowledge on that?
No.
Perhaps that can also help if you, if you get yourself attached a bit more to one of the Caraceae. Robert, you wanted to ask something.
Yeah, a simple question. I must say I'm fairly unfamiliar with Nitella. I just was looking this text up. I would be curious, I mean, as you are comparing 2 living organisms as providing different substrates and possibly affecting the communities living on them. Can you say something how the environment is selecting for Cara versus Nitella? I mean, are there some systematic differences in which environment I find Cara versus Nitella in terms of, for example, nutrient status of the water? Or are they in highly similar water bodies?
I believe that the Cara and Nitella have the same strategies, and the environment could select both in the same form. I don't know. But I believe that the future environment is the same for both macroalgae.
Okay, so Luke had a question because he raised his hand.
Yes, I have one. Thanks, Irma, for your nice talk. I was just a question out of curiosity. You're the second person in the row today that mentioned the word complementarity. And when I think about complementarity, I'm always thinking about the biodiversity ecosystem functions studies that looked at complementarity versus selection effects. And I have sort of the feeling that your definition of complementarity is different. And so I wonder whether you had thought about it and what it actually means, because you could, to some degree, say that what you call complementarity is simply differences. It's simply niche differences or whatever. So can you explain that a bit? Because I find it intriguing that this is the same term. Sorry.
Yeah, when I used the term complementarity, I thought about sorry. Because when I thought macroalgae and epiphytic algae have the same strategy, ecological strategy. So epiphytic algae depends of the macroalgae substrate, you know, and Caraceae are macroalgae and have the same strategies. And I believe the complementarity could display in the coexistence with both communities, epiphytic algae and macroalgae. I don't know if you understand me.
Yeah, but I can imagine that the two plants occur in different patches.
Yeah.
And then you would say they complement each other at the regional scale, but not so much at the very local scale. So there is some difference there, and it's perhaps useful to make that clear, because it could also be confusing to people if you call it just.
Okay.
So it's basically species turnover across an environmental gradient or something like that. And you try to understand why, which is very good. Mm-hmm. But the word complementarity is a bit, difficult. I'm not saying it's wrong. I mean, it's just that it's a different meaning from what many people may have in mind.
Okay. Mm-hmm.
I just wanted to make that comment, but very nice work, by the way.
Thank you.
So thanks, Luke. Any more questions? And actually, we are kind of at the end of the session. So if you find yourself that you might have had a question to any of the previous talks, then it's your time to perhaps shoot again. If not, then I actually have a general question to the 6 presenters of today. I know that David is a very young postdoc, but are all the other students, so all the other 5 presenters? If not, then please raise your hand. If not, yeah, okay, then it means that we have 5 students in this session. That's what I wanted to know, because we have to nominate student talks for the best student presentation, and for that reason we need to know this. So Ryan said she already finished her PhD. Right, okay, then we have 4 possible talks for this. Thank you. So any other question from the audience? I'm checking just the chat. No. So then any final thoughts, Robert, perhaps, before we wrap it up?
Not, but I was really pleased about the variety of systems that have been discussed, and I enjoyed a lot listening to these very different studies. And yeah, I think it's quite inspiring to see different habitat types, and as we have in the title of the session, that all of them in some way are suitable for this approach of thinking about ponds and shallow lakes as highly replicated systems in the landscape that are very amendable for local versus spatial analysis of diversity, community composition. But that's not bringing something new, I think.
Yeah, also thanks from my side. I think it's always a challenge, and now I think with this big conference, it's even more of a challenge to now get adapted to these kind of new forms of presentations. And I want this to be acknowledged, especially because we have so many students in this session, and it's always stressful to present your work. And I think you all did a really good job, and be proud of yourself, because these are really yeah, tiring ways of communicating your data, but you really did a good job.
Exactly. Yeah. At the same time, I think it's a nice way to connect because, I mean, realizing that we are working at very different time. I think Patrick was on the early end and Europe on the late end here. It's a different way of getting together, but it's also, I mean, I think it shows that we all try to do our best. And as Zsófia said, I think you all did a really good job. And yeah, the thing to learn is that it's no need to be shy, no need to be shy to speak up, but just, I think you all contributed that we do the very best in this awkward situation. Thank you.
I would really also encourage you to go to the other sessions, ask questions. Also, if you're a bit perhaps shy to ask it like directly, then use the chat, use the forum. Everybody will be happy for your questions. So just go ahead, be brave. And yeah, enjoy the rest of the conference.
Can I just add something before you guys close the room?
Yeah.
I would like to thank you guys again. This was great for leading everything. I would like to thank Luke because he also helped a lot with the questions and all the participants, all the students. And following up on what Zsófia has just said, I would like to invite everybody, not just for the sessions tomorrow, but for the workshop this afternoon, which starts in about an hour and 5 minutes. The workshop has been designed specifically to address some of these questions, some of these issues that the early career researchers have, but everybody else is invited. I'm gonna copy the link to the meeting here. And even if you're not registered, just join us. And what else? Oh, I should also apologize to Patrick. I'm really sorry about the time there. We couldn't really make it a good time for everybody in the world, but thank you all for joining. And I really hope to see you guys later today in our workshop, okay? Thank you, Zsófia and Robert, once more.
Welcome.
Thank you, bye.
Bye, everyone.
Thanks and bye-bye.